Hello friends, welcome to Department of Plant Pathology, KDSP Agriculture College Nasik. In previous lecture, we have studied all the diseases of mustard, their causal organisms and symptomatology and their management. Today we are going to see the diseases of gram and linseed and their management. Now, first, that is wilt. Wilt of gram is caused due to Fusarium oxysporum forma specialis ciceri. As you can see the symptoms of this disease in this image, there is completely wilting of seedlings will occur in severe infection of this wilt disease. The disease can affect the crop at any stage of crop growth. The field symptoms of wilt are dead seedlings or adult plants and usually occurs in big patches. At seedling stage, that means 3 to 5 weeks after sowing, whole seedlings collapse and lie flat on the ground with dull green leaves and shrunken stem. This is the particular marks of identification for wilt disease. Then dark brown or dark discoloration of the internal stem tissues is also found in severe infection. At adult stage, drooping of petioles, rachis and leaflets, and finally entire plants will die due to the infection of this Fusarium oxysporum. This is all about symptomatology of this Fusarium wilt. Now we will see the management of this Fusarium wilt. For management of Fusarium wilt, we have to apply all integrated disease management practices. That first one is deep summer plowing. Deep summer plowing helps to expose all the chlamydospores which were present inside the soil to the sun's heat, and the initial inoculum may be reduced due to this practice. The next point is follow crop rotation if possible. This will help to break the life cycle of this Fusarium oxysporum. Third point is always use disease-free seeds. That is, seeds used for sowing should be obtained from authenticated sources. Then most important point is seed treatment. Seed treatment should be done with Trichoderma viride at the rate of 4 gram per kg of seeds, or Pseudomonas fluorescens at the rate of 10 gram per kg of seed, or you can also use the chemical seed treatment like carbendazim or thyram at the rate of 2 gram per kg of seed. Similarly, we can also control this disease by using fungicidal drenching with carbendazim or propiconazole as an eradicative method. So for that, we can use 2 gram per liter of fungicidal solution, and such fungicidal solution should be drenched near the rhizosphere soil of this gram crop, so that all the chlamydospores and all the mycelial content or mycelial network which is present in that rhizosphere soil will be killed due to the fungicidal activity of these fungicides. So this is all about management of Fusarium wilt. Next important disease is Botrytis grey mold, and it is caused due to Botrytis cinerea. You can see the symptoms of this Botrytis grey mold. You can easily find out that lack of pod setting is observed in severe infection of this Botrytis grey mold. Under favorable conditions, foliage shows symptoms and plants often die in patches. There are big patches formed due to the infection, and all the foliage should be dropped down. Then shading of flowers and leaves also occur, and covered with spore mass which can be easily seen. Lesions on the stems are 10 to 30 mm long and girdle the stem fully and covered all stem parts. Tender branches break off at the point where the infection occurs, and the grey mold has causes rotting on the infected portion. Affected flowers turn into a rotted mass. Lesions on the pod are water soaked and symptoms. On infected plants, the pods contain either small shriveled seeds or at almost not development of seed occurs in infection. Now we will see the management of this botrytis gray mold. For management of this botrytis gray mold, we have to avoid excessive vegetative growth. That means if you apply high doses of nitrogenous fertilizer, there will be development of canopial growth, and this canopial growth leads to the development of this botrytis gray mold. So that this practice should be followed, that excessive vegetative growth should not be excess. Then intercrop with linseed is found effective for management of this botrytis gray mold. Avoid excess irrigation, use compact varieties. Then deep summer plowing is helpful for breaking the life cycle of this pathogen. Similarly, if we do the seed treatment with carbendazim or thyrum at the rate of 3 gram per kg at the time of or before the sowing, it will reduce the initial inoculum, or it may be avoid the chances of infection of this botrytis gray mold. As a eradicative method, we can also control this disease by spraying with fungicides like mancozeb or bavistine. These are some fungicides which were found effective for management of this botrytis gray mold. The next disease is Ascochyta blight, and the causal organism is Ascochyta rabi. You can see the symptoms of this Ascochyta blight. All plant parts are affected due to this Ascochyta rabi. Symptoms appear on leaves as initially it shows water-soaked lesions. Symptoms include small circular brown spots on the leaves. Under favorable conditions, these spots enlarge rapidly and coalesce and form big patches and shows blighted appearance. That's why it is called as that's why it is called as Ascochyta blight. In case of severe infection, the entire plant dries up suddenly. This is the particular marks of identification of this Ascochyta blight. The lesions are also developed on stems, petioles, and let infection results in shriveling and infected seeds where also occurs. This Ascochyta blight disease is seed-borne disease, and for management of such seed-borne disease, we have to use disease-free seeds for sowing, then follow crop rotation, intercropping with wheat, barley or mustard to break the life cycle of this pathogen. Similarly, seed treatment should be done with carbendazim at the rate of 2 gram per kg of seed, and if the emergence of this disease after sowing, then you can also control it by spraying with mancozeb or with wettable sulfur at the rate of 3 gram per liter of water. This is all about management of this Ascochyta blight. Now we will see the next crop, that is lentil. In lentil, there is common disease, that is wilt, and its causal organism is Fusarium oxysporum forma specialis lentis. Symptoms of wilt of lentil are equal to symptoms of grams, as we have seen in this lecture, that is development of chlamydospore inside the soil, all seedlings were die prematurely and big patches were found, discoloration of vascular bundle. All the symptoms are common and you can refer in the first slide of this video. And management practices are also same as we have seen in wilt of chickpea. So we will move on the next disease, that is rust of lentil. Rust of lentil is caused due to Euromyces crassi or Euromyces vca. These are the causal organism responsible for rust of lentil. The first symptom of this rust of lentil can be seen on leaves. The first sign of the infection is small white pustules which were developed on the lower leaves and upper leaves also. Then minute yellowish slightly raised pustules were developed on leaves that they enlarge, change to orange color in later phases, and these pustules were surrounded by light colored halo. These are some marks of identification for rust of lentil. These rust spores are small, light and may survive for several days. They can be spread to large distance by wind or air currents. Warm temperature, that is more than 20 degrees Celsius, is favorable for development of this rust of linseed. This is all about symptomatology of this rust of lentil. Now we will see the management of this rust of lentil. For management of this disease, we have to use disease-free seeds, follow crop rotation to break the life cycle of the pathogen. Intercropping should be done with wheat, barley and mustard. Seed treatment should be done as we have seen in earlier slide. Similarly, you can also spray the chemicals like mancozeb or bavestin at the rate of 2 gram per liter. In that way, we can control this rust of lentil in very effective manner. Thanks for watching this video.